Hey guys, welcome to the Bio25 cell and molecular biology review session. This is basically review session part 2 of week 6, where we're going to talk about the rest of the pathways from this week's content. And yeah, let's get started. So first pathway I'm going to talk about is the survival pathway. And for the survival pathway, these are just some key ideas to think about, or key questions or so. So some questions I had was, what's the end result of the pathway? What's the pathway inhibit? Describe how survival signaling pathway works. And are you able to explain all the steps in this pathway to someone who's never been in this class? And lastly, how could you cause cell death to occur even with the signal present? So those are just questions I thought, or key concepts to keep in mind just when looking at this pathway. So firstly, I thought it'd be useful to understand what does the pathway do, or what is the cell response of the pathway, or what is the end result of the pathway. And so essentially the end result of this pathway is survival of the cell, hence the name survival pathway. And so since we know that, then we know that this pathway, in order to promote cell survival, that it has to inhibit cell death, which is called cell apoptosis. And actually programmed cell death is the cell's default setting. So by running through this pathway, the cell is able to prevent cell death and allow for cell survival. And another thing I thought would be good to mention is the pathway is important to researchers and biologists, and researchers who study cancer can target this pathway. And this makes sense intuitively because cancer cells replicate at a very high rate, and there's lots of crazy cell division that occurs. So manipulating some of the components of the pathway that could stop cancer cells from dividing and surviving would be beneficial because we want them to stop proliferating. And the pathway works with other pathways to help the cell survive. And you can see from the diagram in the right, I think this is just something off of Google. We have looks like it has like the growth factor pathway, survival pathway, and then you can also see like the GRB2 SOS RAS pathway, which is the RTK MAPK pathway, I believe. But you can just see that there's like a huge group of other proteins that are here. And this is the survival pathway proteins, if you can recognize some of them. But the pathway is in communication with other pathways, obviously, to work together to get the end result of survival. And yeah, that's pretty much the purpose of the signaling pathway. And so now I'm going to just talk through the components of the pathway, so how it works sequentially. And I split up the slides in smaller chunks so that you guys can see like the different steps involved rather than one big large diagram. I just figured it would, breaking it down makes it much easier. But I also have like the entire diagram of it on the next few slides so you guys can see it as a whole. But yeah, so essentially the first thing that will happen is you have the survival signal that binds to the receptor. And this survival signal appears in the form of a dimer, and it will bind to the RTK. And this binding causes the RTKs to dimerize. And the dimerization of the RTK happens, and this enables the RTKs to cross-phosphorylate each other. And then once that occurs, your RTK becomes active. And once the RTK is active, then you will have a protein called PI3 kinase that will bind to the SH2 domain on the RTK, which is this phosphorylated tyrosine here. And it will bind to this site on the RTK. And essentially what happens is the PI3 kinase will phosphorylate an inositol phospholipid, which is this molecule up here. And an inositol phospholipid is, I don't believe, it's not a protein. It's actually a lipid. So inositol phospholipid is this molecule that's, or this lipid that's anchored to the cell membrane. So what will happen is once a PI3 kinase is active, it will phosphorylate an inositol phospholipid, which at this point is in the diphosphorylated state, or it just has two phosphates. And once it gets phosphorylated by PI3 kinase, inositol phospholipid becomes triphosphorylated, so it gets an extra phosphate added to it. And once it's in the triphosphorylated state, the inositol phospholipid becomes active, and it is able to bind to other proteins, which we'll get to in the next slide. And yeah, so that's like the first portion of the pathway. So does anyone have any questions on that so far? You can feel free to unmute yourself or put it in the chat just to make sure that there are no questions before I keep going. Tina, can you say anything in the chat? Because I can see. Oh, no.
Yeah, I think everyone's good.
Solid. Okay. Thanks, Haley. So that's the first part of this, of the survival signaling pathway. So now let's just keep in mind that now we have triphosphorylated inositol phospholipid, or the inositol phospholipid has become active. So what will happen is the next portion. And so this inositol phospholipid, which is now in the triphosphorylated state, will, well, it's actually anchored into the membrane. So that means that it's like restricted in its movement. So what will happen is that there's multiple inositol phospholipids in the cell. So like AKT will bind to the inositol phospholipid, and so will this other protein called protein kinase 1, which can sometimes be abbreviated as PK1. And so you just have to picture this in the cell, that there's more than one inositol phospholipid. So what will happen next is that you'll have AKT will find the inositol phospholipid, and it will bind to it. And the way that AKT is able to bind to the inositol phospholipid is because it has to be, the inositol phospholipid has to be in a triphosphorylated state because it can fit into the binding site of AKT, and this binding site is called the pH domain. So anytime an inositol phospholipid binds to something, that is at the pH domain. So that's just like a little side note. So anyways, what happens next is your inositol phospholipid will bind to AKT, and another one will bind to protein kinase 1. But at this point, AKT is not active yet. So what will happen is AKT will bind to phosphorylated inositol, and then in order for AKT to become active, it has to be phosphorylated at 2 sites. So what will happen is you'll have protein kinase 1 that will come in and phosphorylate AKT at one side of it. And once it's phosphorylated at this site, it causes a conformational shift or shape change in AKT, which exposes the amino acids on its surface so that the other protein kinase 2, is able to find the binding site, and it can bind to AKT at the second site. So basically what happens is these protein kinases, like one at a time, will phosphorylate AKT. And once it binds and phosphorylates AKT, both of these proteins have to do so in order for AKT to become activated. So let me see if I can say it like in shorter terms. So basically what happens at this step is we're trying to get AKT to become active. And that's done so by the help of two protein kinases. So protein kinase 1 will activate AKT on the left. And once that happens, then a conformational shift or shape change happens in AKT, which causes the binding site to become which causes this binding site to appear exposed on its surface so that the protein kinase 2 can bind to AKT at the second binding site. And so that's what we mean when we say like the subsequently phosphorylating AKT, because it has to be one at a time. And both of these proteins have to phosphorylate AKT in order for it to become active. So it can't just be active based off of one protein kinase. It needs both of these to become active. And AKT also is bound to an inositol phospholipid. So yeah, so AKT basically has to be bound to the inositol phospholipid, and the protein kinases have to subsequently phosphorylate it in order for it to become active. And then once AKT is active at this point, it will dissociate from the anchored inositol phospholipid and it will float down to the cytosol to do its job. And that is the next part of the pathway. Does anyone have any questions on what I said so far? It seems like it was a lot, and I just want to make sure people understood.
I think they're good so far. I said that they could type in the chat if they had any questions too, and there's nothing there.
Yeah. I'm just going to say it one more time just because I feel like I kind of confused myself a little. But basically, sorry guys, basically in order for AKT to become active, it will bind to inositol phospholipid in its triphosphorylated state. This binding occurs at the pH domain of AKT. Then once that happens, you'll have protein kinase 1 that will come and phosphorylate AKT at one site. And then that event or phosphorylation event causes a shapeshift in AKT, which exposes this site number 2 for protein kinase 2 to come in and phosphorylate AKT. So once AKT is phosphorylated twice at the two binding sites, then it's able to become active. And then when AKT is active, it will dissociate from the phospholipid inositol, and it will just float down to the cytosol, which is the next step. And yeah, I don't know. I just felt like that time I explained it a bit clearer. But another thing I wanted to mention, which you guys won't see like on the pathway, but it's just good to know, is that there's this special protein called PTEN, which is a phosphatase. And phosphatases essentially remove phosphate groups. So it might come up, it might not, but sometimes Laura will ask questions about what would happen if I had a phosphatase that removed phosphate groups from an inositol phospholipid, or it can even remove phosphate groups off of proteins as well. So PTEN is just a phosphatase, which means it has the ability to remove phosphate from an inositol phospholipid. And so we know that if you were to remove a phosphate group from an inositol phospholipid, it'll become diphosphorylated, meaning that there are only two phosphate groups. And so you could predict that that would affect the function of the pathway in some way, shape, or form. And yeah, it's not pictured in the pathway, but it's just a good thing to keep in mind. Cool. So can I move to the next slide? Does anyone have questions on that? I'm going to take that as no, and I'll just keep going. So now the next slide is going to talk about the next section of the pathway, which is what happens when we have activated AKT and it dissociates from all this stuff and goes to the cytosol. So that is the next step. So now we have activated AKT, and as you can see, it's dissociated from the protein kinases and the inositol phospholipid. So now it's floating in the cytosol. So what will happen is AKT is pretty important for this part. So activated AKT will bind to this protein called BAD. And what will happen is the AKT will phosphorylate BAD, which will cause BAD to become inactive or deactivated. And so this might be a bit tricky to understand because I feel like up to this point, you guys have only seen phosphorylation as a method of activating proteins, but phosphorylation can also be used to deactivate things. So just keep in mind that active AKT, when it phosphorylates bad by interacting with it, causes bad to become inactive. And so this is important because normally before AKT comes along, bad as a protein, when it's active, it's bound to this special protein called BCL2. And BCL2 is an important protein, which I'll get to in the next slide, because BCL2 is an apoptosis inhibitory protein. And apoptosis inhibitory protein means essentially is that it's responsible for inhibiting cell death. And so what normally happens is when BAD is active, it's permanently bound to this BCL2 protein. And that basically means that this BCL protein is inactive and it's unable to do its function of preventing cell death. And so what will happen is when AKT comes along and phosphorylates BAD, causes BAD to become inactive. And the inactive form of BAD will actually bind to a different protein. So you don't need to know this protein's name, but just know that when BAD is inactive, it no longer binds to BCL2. And so this is a good thing for this pathway because when active AKT comes along and phosphorylates BAD, it makes BAD inactive. And BAD is holding BCL2 in captivity. And so essentially, when BAD is inactivated via the phosphorylation of AK2, this means that the BCL2 protein is now free in the cell to go and do its job. And the BCL2 job is to inhibit cell death. So to stop cell death from happening, which means, in other words, meaning that promoting cell survival. And so basically, you can think of it as like BAD is blocking the protein that blocks cell death. It's BAD is inhibiting the inhibitor, which sounds kind of silly, but it's basically BAD is blocking the protein that's responsible for helping a cell survive. So when BAD is inactive, this protein is free to go do its job and save the cell. And so essentially, that's what I mean here when I say BCL2 is free to actively prevent programmed cell death. And it does so by binding to mitochondria, but I don't think she focuses on this detail. I just thought it was good to include. And so, yeah, so the end result of the pathway is AKT phosphorylates BAD, causes it to become inactive, and the BCL2 protein is free to go and prevent cell death because it's an apoptosis inhibitor, which means it's a cell death inhibitor, which means it's a survival promoter or survival, which means that essentially if it's inhibiting cell death, it means that it's just promoting survival. So yeah, I know that was a lot, but I also have more slides on this to help it make sense. Does anybody have any questions on what I explained so far? I'm going to take that as no, and I'm going to keep going. So essentially, I figured I'd explain it a few more times just to help it make a bit more sense. And so basically, in this pathway, the default response or the default setting is cell death, which is called apoptosis. And we have this protein BAD, which is a protein that initiates apoptosis, which means it allows it to happen. And we have this protein BCL2, which is bound to BAD when BAD is active, which stops cell death from happening. And so basically, when you have BAD that's active, it's clinging on to BCL2. And when BAD is bound to BCL2, then BCL2 is inactive and it's unable to do its job of promoting cell survival. And so I just put a real life example of you get mitochondrial holes. And so that leads to apoptosis. And so the flip side of that is when the signal survival signaling pathway is working, and active AKT comes along and phosphorylates bad, which makes it inactive. And then when bad is inactive, it's not bound to BCL2 anymore. And so BCL2 is not bound to anything. So then it is active and it's free to go and inhibit apoptosis. And inhibiting apoptosis leads to cell survival. So the cell lives. And so, yeah, that's basically what I mean when I say that bad inhibits the inhibitor. So if you inhibit the inhibitor of cell death, then you get cell death. But if you don't, if you stop BCL2 from being inactivated, then you get cell survival. So does that make a bit more sense for people in terms of how bad and BCL2 works? Because I know that could be tricky. I'm going to take that as a yes, because I didn't get questions. Hold on. That was helpful? Okay, thanks. Thanks, Haley. Cool. And because I like diagrams, I have a diagram of everything I just said, which is basically the two scenarios, right? So when bad is active, it is blocking BCL2. And when you block BCL2, you inhibit the inhibitor. So if you inhibit the inhibitor of cell death, then you get, if you inhibit the inhibitor of cell survival, I'm mixing it up now. I'm sorry. If you inhibit the apoptosis inhibitory protein, you get cell death. Sorry, that's kind of a mouthful to say. And then the second scenario shows what happens in our regular pathway, which is, you have the active AKT, which phosphorylates bad, makes bad inactive. Then BCL2 is kind of free to go and save the day, which results in blocking apoptosis and also known as cell survival. So hopefully that makes sense. I just showing it in different forms like words and diagrams helped me to make it make sense. So I hope that it makes a bit more sense what happens, because I know it can be confusing with the phosphorylation in activating a protein. So yeah, that's just the explanation of that. Does anyone have any questions at all? Let me peep the chat. I can't see the chat.
There's nothing in the chat either. Yeah.
Okay, cool. So yeah, like I said, you have this slide here, which is essentially the entire pathway I just explained, but I broke it down into small little steps. I just think it's for explaining purposes, it's easier to explain it that way. But here's the big picture pathway. Highly recommend you guys go and you practice walking through the steps yourself. Using a blank diagram like this, you can kind of quiz yourself and say, okay, this happens sequentially in this order, then this happens, because pathways have a chronological flow. So it's important to understand what happens in a pathway normally when things are normal, so that you guys are prepared to answer questions about what could possibly happen if, something goes wrong, or if we mutate a protein, or if we change something in the pathway. Because pathway deviations and mutations and things going wrong is the types of questions that you would be asked by Laura on exams or problem sets. So yeah, so definitely recommend practicing this. You can explain to a goldfish, your friend who doesn't take this class, or parents, whoever. Just kind of say it in your head or in the shower or something, just so based off of this blank picture with no notes, if you can understand how the pathway works, or you can explain it to someone else, then you have a solid grasp of it. So yeah, that's basically why I have the blank picture with everything, so you can see it, the big picture of the entire pathway, and just so you guys can practice. So I hope that helps. And Yeah, and this slide is basically everything we just went over, so I don't feel the need to repeat it. But I know people have different learning styles. So some people like to see a whole list of everything that happens. Some people like the small steps, like pathway broken up into smaller chunks. And then some people just like to look at the empty pathway and try to figure everything out, like how it happens in their head. So just thought I would cater to the different learning styles and put a list of stuff. But it's basically just a summary of everything I said. So I'm gonna just skip this slide unless people have questions because it's everything I said and probably a little more detailed, but yeah, everything I said. So, oh, I don't know why I have the picture again, but there's the picture. And yeah, so now I'm gonna jump to the next pathway that we'll talk about. This one is called the JAK-STAT pathway. Main ideas for this pathway is, or main questions, I should say, is the JAK-STAT pathway considered to be an RTK, receptor tyrosine kinase? And then how does the JAK-STAT pathway work? And can you explain this pathway to someone who's never taken the class before? Okay, and so just in case you guys were not sure what a receptor tyrosine kinase is, I figured I'd put a recap of what it is in the slides. This is also from the Wednesday review session. So essentially, RTKs have three parts. They have the extracellular binding domain and the intracellular portion, which is made of like the transmembrane portion and the part that's inside the cytosol. So that's RTKs. And then just the way RTKs work, we know this based off of like the survival pathway as well, is you just have a signaling molecule that comes in the form of a dimer, binds to the two RTK monomers. The RTKs come close in proximity, or in other words, dimerize. And this dimerization causes the stimulation of kinase activity. And then once the kinase activity is stimulated, the RTK monomers are able to cross-phosphorylate each other and in the intracellular portion. And this phosphorylation of the RTK or the active RTKs create binding sites for other downstream proteins. So that's just the basic mechanism of how RTKs work. So actually, I forgot to ask. So based off of that, can anyone tell me whether they think that the JAK-STAT, the JAK-STATs or the receptors from the JAK-STAT pathways are considered to be RTKs? Or can anyone take a guess for me?
I would guess that they wouldn't be RTKs because we need the two JAKs to phosphorylate the intracellular receptor portion after it's been dimerized.
Yeah, so you're actually correct. So the JAK-STATs would not be considered to be RTKs. And I think you're on the right track for the reasoning. So let me see if I can explain it. And I think you're definitely on the right track. I think you might actually have nailed it. So let me explain it and then let me see if what you said was the same thing. So Ethan was right. So the JAK-STAT pathway does not have RTKs, and the JAK receptor itself is not a RTK. It's still a receptor, but since it does not have a kinase domain, it doesn't have this autophosphorylation function. And so it's not like the RTKs where the receptors can cross-phosphorylate each other and they have like built-in autophosphorylation mechanism. And so the JAK-STAT pathway has these special prolactin receptors that just has proteins tacked onto them. And these proteins are called JAKs. So you see in the diagram, you have JAK1 and JAK2. And what is doing the phosphorylation here is not necessarily the receptor, it is the JAK proteins themselves that do the cross-phosphorylating in this pathway. So that autophosphorylation activity is not within the receptors itself, but it's these proteins that are able to phosphorylate each other. And the cross-phosphorylation of each JAK-STAT protein is what causes them to ultimately become active. And it's not considered an RTK because the entire receptor itself is not doing this autophosphorylation. It's just like a receptor that happens to have these two proteins tacked onto them. And yeah, so that's the gist of that concept. And JAK-STAT pathway is pretty important because it's involved in immune system development and memory gland development and lactation. And so that's the purpose of this pathway. It produces proteins involved in milk production. And I think it's specifically for breastfeeding. And so just like I said before, JAK-STAT pathway has receptors, but these receptors are not RTKs because the receptor itself does not have a kinase domain. And it has kinase subunits, or these kinase proteins that are kinases, JAK1 and JAK2, that associate with it. So yeah, does anyone have any questions on this page? And so no questions. So I'm just gonna real quick say something else. So JAKs are proteins. So JAK stands for just another kinase, at least when the scientists were naming this. So if that helps you remember that these proteins, these ones in blue, are kinases, and they're the ones with kinase activity and not the receptor itself, then that's a good thing. The name itself should be able to help. So JAK is just another kinase. I think it's also called Janus kinase. But yeah, it's a non-receptor tyrosine kinase. I said it 3 times, so hopefully you guys understand why it's not an RTK. But yeah, I'm just gonna go to the next slide if there are no questions. And Tina, you can actually continue off this next half if you'd like.
All right, so I'm gonna go into a little more detail about like how the JAK-STAT pathway actually occurs. So like Michelle said, there's a bunch of different signaling outcomes that JAK-STAT can do, but the example that Laura explained or talked about during the lecture is specifically known for milk protein production in breast milk, or it's known as lactation. So basically what occurs is you have JAK1 and JAK2 bound to the receptor, those blue circles bound to the green legs, I guess. And then you have this prolactin signal that comes in to bind to the receptors, and that causes the 2 green receptors to dimerize and come together. When the 2 green receptors come together, that brings the JAKs closer together. And so it can activate each other by cross-phosphorylating each other. So JAK1 activates JAK2, JAK2 phosphorylates JAK1, cross-phosphorylation. And once those JAKs are activated, it can go and then activate the receptor itself. So that's causing those phosphatases that Michelle's pointing at right now, those bottom P circles. So yeah, so then now the receptor is activated and it has these binding sites for STAT monomers to come in and bind to those phosphotyrosine locations. And it can do so because the STATs have SH2 domains. And SH2 domains, just a reminder, they are found in many signaling molecules, and they're just phosphotyrosine binding sites. So the SH2 domain of STATs bind to the phosphorylated receptors, and then the JAKs above the STATs can go and phosphorylate the STATs. So like that pink arrow that Michelle's pointing out. Once the STATs are further phosphorylated, or not further phosphorylated, once the STATs are phosphorylated by the JAKs, they go through a conformational change that allows the STATs to disassociate from the receptors. And so once they disassociate their respective SH2 domains, they kind of like flip, and then they dimerize together to form that dimerization, the active STAT5 dimer that Michelle's circling. And then from there, now that it's active, it's a dimer, it can then go and enter the nucleus via important, as we've learned before, that mechanism a couple of weeks ago, and it'll act as a transcription factor where other transcription factors and regulators can come and bind, and essentially it'll go and signal downstream for transcription of the milk protein genes. Oh, okay, yeah, we can go to the next one if no one has any questions.
Oh yeah, I just wanted to add that the STAT5 functions as a protein and as a transcription factor. So that's how it's able to sort of like make its way into the nucleus once it becomes active, the active STAT5 dimer. And just like Tina said, don't forget your SH2 domains. Anytime you see phosphorylated tyrosine sites, the STAT5 dimer itself, like how the STAT proteins are able to connect to each other, is they intermingle through the SH2 domains. And PEEP, the SH2 domain that is found on the receptor, the prolactin receptor, which is what enables the STAT5 proteins to bind there in the first place. And yeah, and so once the SH2 domain is available, the STAT5 can bind there, and then it gets phosphorylated because it's close in proximity to the JAK proteins. And so that's what enables it to dissociate from the receptor. So yeah, I just thought I'd point that out. But yeah, we can go to the next one.
All right. So then this is just another way, basically everything that Michelle and I just said in that last slide, just a more like physically written out for you guys in case some people prefer the written version. But again, the JAK-STAT pathway receptor is a heterodimer. It has the receptor portion where the receptor binding domain is extracellular outside of the cell, and then the JAK itself is a non-receptor tyrosine kinase, again, just to kind of drill that into your heads. And that is intracellular inside the cell. Then you have the signal that binds to the receptors. The receptors come together and dimerize. That causes the JAKs to cross-phosphorylate via their kinase domains. And then each JAK can then phosphorylate the receptor. Though that allows for binding sites to be available for the STATs to come in and bind to those binding sites, their SH2 domains. The STATs can then become phosphorylated by the JAKs, and then that causes the STATs to dissociate, come together and dimerize themselves. They are activated, they go into the nucleus, and then they cause transcription for whatever needs to be done. Any questions so far about JAK-STAT? All right, guess not. But basically this is again, just a big diagram for you guys to, if you ever want to come back and practice, which is definitely a good way to kind of refresh your own memory or test yourself to know whether or not you actually know every function, because a lot of times exam questions are like a lot of loss of function, gain of function type of ideas. So if you know what it's supposed to do normally, then you can answer questions where Laura will say like, oh, if this breaks or if this gains, anything along those lines, it definitely helps. Any questions about JAK-STAT before we move on to GPCR? All right, if not, we can move on. So the GPCR pathway, again, we're gonna break it down by some like little, not little, but separate portions, and then you guys will be able to see like the big picture after. Oh, we can go on to the next slide. Thank you. So the GPCR stands for G protein coupled receptors. Some of the main ideas that you want to understand about the GPCRs is how does GPCR signaling work, of course. Make sure that you are able to explain all of the steps. We like to say that if you can explain it to someone who's never really taken this class before, has like basic biology knowledge, then you're in a good spot. And then what is the GEF analog in GPCR signaling and what's the GAP analog in GPCR? And basically that just means that what acts or is like a GEF and what acts or is like a GAP in this situation. And if you don't know that now, we're going to get into that and explain it. And then hopefully by the end of this, you will understand what they are. Okay, so that diagram is how GPCR works. So I'm going to explain it really quick. And then the next slide is like everything written out for you basically, so that you can have that written portion for your own like studying use and such. So GPCRs are made of a 7 transmembrane receptor that's bound to a G protein, and that G protein is made of 3 subunits, the G alpha, beta, and gamma. So it's called a 7 transmembrane because it goes through the membrane 7 times. So that's why it's kind of like a squiggly receptor. And then the G protein is the blue, red, and green portion. All together, that makes the G protein. It just has 3 subunits. So that's why it's called known as a heterotrimeric complex. And then the G alpha is actually a GTPase. And so that'll come more, that's good knowledge for down the line of this signaling pathway. So in the inactive state, the GPCR is bound to the G protein and the G alpha has GDP attached to it. When a ligand comes in to bind to the GPCR, that'll cause the G alpha to let go of its GDP. And then due to just a lot of GTP around in the cell, GTP can easily diffuse into now that open binding spot of G alpha. And once G alpha has GTP, it can become activated. So once G alpha is activated, this will then cause G beta and G gamma to dissociate. So G beta and G gamma are together, and then G alpha is its own thing. It goes off and it kind of acts on its own, but G beta and G gamma will stay together, and then G alpha will be on its own. But once G alpha is activated with GTP, it'll dissociate from beta and gamma. And so because of that, you can guess now that the GPCR itself, that receptor is the GEF analog, because it's what's causing that exchange when the ligand binds for GDP to go out and then GTP to come in. So that's what's the analog for GEF. But continuing down the signaling pathway, now that G alpha is active, it can go downstream and bind to different effectors to cause a signaling transduction cascade. So G protein coupled receptors, they're really versatile. They are, as Laura has explained many times, like there's so many different types of them, like 800 plus or something like that. And basically the essential mechanism is all the same, where it'll just go downstream to further activate and cause a signal transduction cascade of whatever needs to be done for whatever product the cell needs to get. And then once the cell has what it needs and activation has all occurred, all the signaling that's occurred has happened, it needs to deactivate, or if it does deactivate, the G alpha in itself, there's different ways that G alpha can be hydrolyzed so that it goes back to GDP for it to be inactive. But one of the ways would be like G alpha itself has intrinsic rate of hydrolysis, but it's usually not the most effective because it's not necessarily the fastest. But so a better way would be for RGS to bind to G alpha, and then that'll cause G alpha's intrinsic GTP activity to accelerate, causing GTP to hydrolyze to GDP more easily. So that's kind of, you can see RGS is like that light blue color on the left right there. And then once that entire complex is deactivated, G alpha will diffuse back with beta and gamma, becoming a heterotrimeric complex again. And then as a whole, that deactivated complex will go and bind back to the GPCR receptor. If that ligand is still there, it'll just a cycle become activated again. If that ligand is no longer there, it'll all quiet down, become deactivated together until a ligand comes back again, and then the cycle starts again. So that was definitely a lot. Does anyone have any questions? Yeah, this is like written out in like words so that you guys, if you want to read it really quick, have any questions.
I actually do have a question.
Yeah, of course.
So when the G alpha dissociates from G beta and G gamma,
Mhm.
both G alpha and the G beta G gamma complex can then go to effector proteins and send separate signals.
Yeah, so G alpha goes to do G alpha is kind of like a lone ranger. That's how I think of it. It goes
Okay.
down to do whatever job that G alpha needs to do. And then G beta and G gamma are a duo where they stay together, and then they'll go kind of like the other way or just in a different direction to cause a signal transduction of their own. And it's important to remember that they don't just like disperse around the cell. They have a like a lipid, I can't remember what it's called, but it's like a lipid attachment to the cell membrane itself. So it's like diffusing across the not across the membrane, along the membrane, kind of like they're just going back and forth or moving along the membrane. So that's important to remember, because I know that sometimes people think it like detaches from the membrane, but it's always with the membrane. They just kind of go off to do their own things until G alpha is deactivated by being hydrolyzed, and then it'll diffuse back to the beta and gamma.
Okay. And do G alpha and the G beta G gamma complex, do both of those create the same signal or two different signals?
Michelle, correct me if I'm wrong, but I believe there are two different signals. Like they have different purposes usually.
Can you repeat the question?
So I guess, yeah, what I'm trying to get at is when you have the G alpha that goes off to one effector protein, and then you have the G beta or the gamma beta complex that goes to combine with like another effector protein, are both of those things producing the same signal or two different signals?
I think they're two different signals, but you shouldn't really get tripped up on that portion. Laura's more concerned that you know that once the GPCR is active, just like the function of G alpha. So G alpha
Oh, okay.
dissociates, does its own thing, and eventually it tacks back onto the it's not called heterodimer. I'm losing the word.
Heterotrimeric.
Yeah, heterotrimeric complex once G alpha is done doing its signaling. Yeah, so the beta and the gamma, not worry so much about that. Just know that, once the GPCR is active, G alpha dissociates, does some sort of signaling activity, and then it, at the end of the pathway, it's kind of brought back into the complex here. So does that make sense?
Yeah, thank you.
Yeah, of course.
Does anyone else have any other questions? All right, well, this is just another diagram. It's a little simpler to look at, I would say. That's picture on the side is like a more accurate version of how the GPCR actually looks like. It's not just like a green clump, or not any colored clump, but it's important to remember that it's 7 transmembrane domains, so it goes through 7 times. And so again, there is a signal that binds, and that'll cause the G alpha to exchange GDP for GTP. So thus the G protein coupled receptor that acts as the GEF analog. And then the alpha subunit will dissociate from the gamma and beta subunits, which will trigger some type of cellular response. And then GTP is hydrolyzed back to GDP, either through RGS, most likely through RGS, because that'll help further accelerate the intrinsic activity of the G alpha to hydrolyze. So RGS is like the GAP analog. And it's important to remember too that like GAP isn't what's actually causing the hydrolysis. So RGS itself is not going to cause the hydrolysis. It's just helping the hydrolysis process along and make it go faster. And then after that, the alpha subunit will come back together with the beta and gamma subunits because it's deactivated. And then the cycle starts again.
Yeah, are we good with this? Should I go to the next slide?
Yeah, I think if no one has questions, we're good. All right, and then of course, this is just the big picture diagram for anyone to come back to and just review themselves. Make sure you understand all the different parts, like what each subunit does, what the receptor does, the role of GAPs and GEF analogs, what they are, things like that. Just making sure that you know the different parts of the mechanism so that if Laura asks a question that like breaks the mechanism, then you would understand what will happen and what won't happen. Does anyone have questions about GPCR? All right, well, again, just common misconceptions. I touched on this a little bit already, but the GAP-GEF functions, so they're not enzymes, they help proteins or other components of the pathway. So like I said, the GEF, it'll promote or help along the GDP to dissociate for GTP to diffuse in. And then GAP, it'll help accelerate the GTPase enzymatic activity for hydrolysis, but it's not what's actually causing the hydrolysis itself. So in CAPS, in case you didn't get it, GAP does not hydrolyze GTP, it just assists the enzyme in doing so. And an example, if you want another example, would be like the RAN from nuclear import-export that we learned a couple weeks back. And then remember also that regulation signals and pathways, so everything we've talked about so far, they're all highly regulated, and that's so that there's activation and deactivation regulation so that you just don't have too much or too little of a signal, because of course that's bad for the cell energy and just like production wise. So it'll like prevent, for example, overproliferation where it's like making too much of something, whatever the product of that cell is. And then, yeah, just making sure again that you'll understand all the proteins and their functions, the different components of the mechanism. So if you can understand how it works normally, then you should hopefully be able to better understand or better be able to answer like gain of function, loss of function questions. I think we can go on.
Oh, yeah,
Oh
sorry.
yeah, no, it's all good.
And yeah, that's pretty much it for the review session today. Feel free to reach out to us by email if you have questions. Both Tina and I really encourage you guys to practice this stuff. So definitely try to use the slides that don't have any words on them. I call them wordless slides, so just slides that have the huge images of the pathway, and being able to explain that stuff to yourself or to someone else will help you understand what's going on. And if you can accurately explain it to someone else, then it shows that you have good understanding. And try to also always keep in mind like ways to shut down the pathway or stop, or what would happen to the pathway if you tampered with certain components. And yeah, that's pretty much all we have for you today.